Chapter 2, Biological Classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological character to classify plants into trees, shrubs, and herbs. He also divided animals into two groups: those which had red blood and those which did not. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdom was developed that included all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi organisms. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification used for a long time was found inadequate. Beside gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, method of reproduction, evolutionary relationship, etc. Classification system for the living organism have hence undergone several changes over the time. Though plant and animal kingdom have been a constant under all different systems, the understanding of what groups organisms be included under this kingdom have been changing. The number and nature of other kingdom have been understood differently by different scientists over the time. Table 2.1, Characteristic of the five kingdom. Characteristic five kingdom: Monera, Protista, Fungi, Plantae, Animalia. Character cell type: Monera prokaryotic, Protista eukaryotic, Fungi eukaryotic, Plantae eukaryotic, Animalia eukaryotic. Cell wall: Monera non-cellulosic polysaccharide plus amino acid, Protista present in some, Fungi present with chitin, Plantae present cellulose, Animalia absent. Nuclear membrane: Monera absent, Protista present, Fungi present, Plantae present, Animalia present. Body organization: Monera cellular, Protista cellular, Fungi multicellular loose tissue, Plantae tissue organ, Animalia tissue organ system. Mode of nutrition: Monera autotrophic, chemosynthetic and photosynthetic, and heterotrophic, saprophytic and parasitic. Protista autotrophic, photosynthetic and heterotrophic. Fungi heterotrophic, saprophytic and parasitic. Plantae autotrophic, photosynthetic. Animalia heterotrophic, holozoic and saprophytic, etc. R.H. Whittaker 1969 proposed a five-kingdom classification. The kingdom defined by him were named Monera, Protista, Fungi, Plantae and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. Table 2.1 gives a comparative account of different characteristics of the five kingdoms. The three-domain system has also been proposed that divide the kingdom Monera into two domains, leaving the remaining eukaryotic kingdom in the third domain and thereby a six-domain classification. You will learn about this system in detail at higher classes. Let us look at this five-kingdom classification to understand the issue and consideration that influence the classification system. Early classification system included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperm and angiosperm under plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their cells. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae cyanobacteria with other groups which were eukaryotic. It also grouped together the unicellular organism and the multicellular one. Say for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group fungi and the autotrophic green plants, though they also showed a characteristic difference in their walls composition. The fungi had chitin. in their walls, while the green plants had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas, chlorella, early placed in algae within plants and both having cell wall, and with paramecium and amoeba, which were early placed in the animal kingdom which lacked cell wall. It has put together organisms which in early classification were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future to depending on the improvement in our understanding of characteristic and evolutionary relationship. Over time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological, and reproductive similarities but is also phylogenetic, that is based on evolutionary relationship. In this chapter, we will study characteristic of kingdom Monera, Protista, and fungi of the Whittaker system of classification. The kingdom Plantae and Animalia, commonly referred to as plant and animal kingdoms respectively, will be dealt separately in chapter 3 and 4. 2.1 Kingdom Monera. Bacteria are the sole member of the kingdom Monera. They are the most abundant microorganism. Bacteria occur almost everywhere. Hundreds of bacteria are present in handful of soil. They also live in extreme habitats such as hot spring, desert, snow, and deep oceans where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under four categories based on their shape: the spherical coccus, plural cocci; the rod-shaped bacillus, plural bacilli; the comma-shaped vibrium, plural vibrio; and the spiral spirillum, plural spirilla. Figure 2.1. Figure 2.1 is showing bacteria of different shapes. Though the bacterial structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrate. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs, that is, they depend on other organisms or on dead organic matter for food. 2.1.1 Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty areas, halophils, hot spring, thermoacidophils, and marshy areas methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and his feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane biogas from the dung of this animal. 2.1.2 Eubacteria. There are thousands of different eubacteria or trubacteria. They are characterized by the presence of a rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as blue-green algae, have chlorophyll A similar to green plants and are photosynthetic autotrophs. Figure 2.2. The cyanobacteria are unicellular, colonial, or filamentous, freshwater, marine, or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts. Example: nostoc and anabena. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrate, nitrites, and ammonia and use the released energy from their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume. Figure 2.2 showing a filamentous blue-green algae nostoc, heterocyst, mucilaginous sheath, roots, etc. Some are pathogens causing damage to human beings, crops, farms, animals and pets. Cholera, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Figure 2.3. Sometimes, under unfavorable conditions, they produce spore. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to the other. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cell known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. Figure 2.3 a dividing bacterium showing cell wall, cell membrane and DNA. 2.2 Kingdom Protista. All single-celled eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What? Maybe a photosynthetic protistian to one biologist, maybe a plant to another. In this book, we include chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoan under Protista. Members of Protista are primarily aquatic. This kingdom form a link with the other dealing with plants, animals, and fungi. Being eukaryotes, the protistian cell body contain a well-defined nucleus and other membrane-bound organelle. Some have flagella or cilia. Protista reproduce asexually and sexually by a process involving cell fusion and zygote formation. 2.2.1 Chrysophytes. This group includes diatoms and golden algae, desmids. They are found in fresh water as well as in marine environment. They are microscopic and float passively in water current plankton. Most of them are photosynthetic. In diatom, the cell wall form two thin overlapping shell which fits together as in a soap box. The walls are embedded with silica and thus the walls are indestructible. Thus, diatoms have left behind large amount of cell wall deposited their habitat. This accumulation over billions of years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing filtration of oil and syrups. Diatoms are the chief producer in the ocean. 2.2.2 Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue or red depending on the main pigment present in their cell. The cell wall has stiff cellulose plate on the outer surface. Most of them have two flagella. One lies longitudinally and the other transversely in a furrow between the wall very often. Red dinoflagellates, example goniolax, undergone such rapid multiplication that they make the sea appear red. Red tides. Toxin released by such large number may even kill other marine animals such as fishes. 2.2.3 Euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of a cell wall, they have a protein-rich layer called pellicle, which make their body flexible. They have two flagella, a short and a long. Though they are photosynthetic in the presence of sunlight, when deprived on sunlight, they behave like heterotrophs by predating on other smaller organisms. Interestingly, the pigment of euglenoids are identical to those present in higher plants, example euglena, figure 2.4b. 2.2.4 Slime molds. Slime molds are saprophytic protists. The body moves along decaying twigs and leaves engulfing organic material under suitable conditions. They form an aggregation called plasmodium, which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiates and forms fruiting bodies, bearing spores and their tips. The spores possesses two walls. They are extremely resistant and survive for many years, even under adverse conditions. The spores are dispersed by air current. 2.2.5 Protozoan. All protozoan are heterotrophs and live as predators on parasite. They are believed to be primitive relative of animal. There are four major groups of protozoan. Amoeboid protozoan. These organisms live in fresh water, seawater or moist soil. They move and capture their prey by putting out pseudopodia, false teeth as in amoeba. Marine forms have silica shell on their surface. Some of them, such as entamoeba, are parasites. Flagellated protozoan, the members of this group are either free living or parasitic. They have flagella. The parasitic form cause disease such as sleeping sickness, example trypanosoma. Ciliated protozoan, these are aquatic actively moving organisms because of the presence of thousands of cilia. They have a cavity gullet that opens to the outside of the cell surface. The coordinated movement of rows of cilia cause the water laden with food to be seated into the gullet. Example paramecium. Figure 2.4d. Sporozoans. This includes diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium, malarial parasite, which causes malaria, a disease which has a staggering effect on human population. 2.3 Kingdom fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. You must have seen fungi on a moist bread and rotten fruits. The common mushroom you eat and toadstool are also fungi. White spots seen on mustard leaves are due to a parasitic fungus. Some unicellular fungi, example yeast, are used to make bread and beer. Other fungi cause disease in plants and animals. Wheat rust causing coccinia is an important example. Some are the source of antibiotics, example penicillium. Fungi are cosmopolitan and occur in air, water, soil, and on animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yes, it is to prevent food from going bad due to bacterial or fungal infection. With the exception of yeast, which are unicellular, fungi are filamentous. Their bodies consist of long, slender, thread-like structure called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tubes filled with multinucleated cytoplasm. These are called sinusoidal hyphae. Others have septae or cross walls in their hyphae. The cell wall of fungi are composed of chitin and polysaccharide. Most fungi are heterotrophic and absorb soluble organic matter from dead substrate and hence are called saprophytes. Those that did not depend on living plants and animals are called parasites. They can also live as symbionts in association with algae as lichen and with roots of higher plants as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission, and budding. Asexual reproduction is by spores called conidia or sporangiospore or zoospore, and sexual reproduction is by oospores, ascospore, and basidiospore. Various spores are produced in distinct structure called fruiting bodies. The sexual cycle involves the following three steps. First, fusion of protoplasm between two motile or non-motile gametes called plasmogamy. Second, fusion of two nuclei are called karyogamy. Meiosis in zygote resulting in haploid spores. When a fungus reproduces sexually, two haploid hyphae or compatible mating types came together and fuse. In some fungi, the fusion of two haploid cells immediately results in diploid cell 2N. However, in other fungi, Ascomycetes and Basidiomycetes, an intervening dikaryotic stage N plus N, that is two nuclei per cell. occur. Such a condition is called a dikaryon, and the phase is called dikaryophase of fungus. Later, the parental nuclei fuse and the cell become diploid. The fungi from fruiting bodies in which reduction division occur, leading to formation of haploid spore. The morphology of the mycelium, mode of spore formation, and fruiting bodies form the basis for the division of the kingdom into various classes. 2.3.1 Phycomycetes. Members of Phycomycetes are found in aquatic habitat and on decaying wood, in moist and damp places, or as obligate parasite on plant. The mycelium is aseptate and cynocytic. Asexual reproduction takes place by zoospore motile or by aplanospore non-motile. These spores are endogenously produced in sporangium. A zygospore is formed by fusion of two gametes. These gametes are similar in morphology, isogamous, or dissimilar, anisogamous or oogamous. Some common examples are mucor, figure 2.5a, rhizopus, the bread mold mentioned earlier, and albugo, the parasitic fungi on mustard. 2.3.2 Ascomycetes, commonly known as sac fungi. The Ascomycetes are mostly multicellular, example penicillium, or rarely unicellular, example yeast. Saccharomyces. They are saprophytic, decomposer, parasitic, or coprophilous, growing on dung. Mycelium. Mycelium is branched and septate. The asexual spores are conidia, produced exogenously on the special mycelium called conidiospores. Conidiophorus. Conidia on germination produce mycelium. Sexual spores are called ascospores, which are produced endogenously in sac-like aci, singular ascus. These are arranged in different types of fruiting bodies called ascocarp. Some examples are aspergillus, figure 2.5b, claviceps, and neurospora. Neurospora is used extensively in biochemical and genetic work. Many members, like morels and truffles, are edible and are considered delicacies. 2.3.3 Basidiomycetes. Commonly known form of Basidiomycetes are mushroom, barked fungi, or puffball. They grow in soil, on logs and trees, stump, and in living plant bodies as parasites, example rust and smut. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about by fission of two vegetative or somatic cells of different strain or genotypes. The resultant structure is dikaryotic, which ultimately give rise to basidium. Karyogamy and meiosis take place in the basidium, producing four basidiospores. The basidiospores are exogenously produced on the basidium, plural basidia. The basidia are arranged in fruiting bodies called basidiocarp. Some common members are agaricus, mushroom, figure 2.5c, Eustyligo smut, and Puccinia, rust fungus. 2.3.4 Deuteromycetes, commonly known as imperfect fungi, because only the asexual or vegetative phase of these fungi are known. When the sexual form of these fungi were discovered, they were moved into classes they rightly belong to. It is also possible that the asexual and vegetative stage have been given one name and placed under Deuteromycetes, and the sexual stage another and placed under another class. Later, when the linkages were established, the fungi were correctly identified and moved out of Deuteromycetes. One perfect sexual stage of member of Deuteromycetes were discovered. They were often moved to Ascomycetes and Basidiomycetes. The Deuteromycetes reproduce only by asexual spores known as conidia. The mycelium is septate and branched. Some members are saprophytes or parasites, while a large number of them are decomposer of litter and help in mineral cycling. Some examples are Alternaria, Coledotrichum, and Trichoderma. 2.4 Kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll containing organisms commonly called plants. A few members are partially heterotrophic such as the insectivorous plant or parasites. Bladderwort and Venus flytrap are examples of insectivorous plant, and Cuscuta is a parasite. The plant cells have an eukaryotic stage with prominent chloroplast and cell wall mainly made up of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plantae includes algae, bryophytes, pteridophytes, gymnosperm and angiosperm. Life cycle of plant has two distinct phases, the diploid sporophytic and the haploid gametophytic. They alternate with each other. The length of the haploid and diploid phase and whether these phases are free living or dependent on other vary among different groups in plants. This phenomenon is called alternation of generation. You will study further details of this kingdom in chapter 3. 2.5 Kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cell-like cell wall. They directly or indirectly depend on plants for food. They digest their food in an internal cavity and store food reserve as glycogen or fat. Their mode of nutrition is holozoic by ingestion of food. They follow a definite growth pattern and grow into adults that have a definite shape and size. Higher forms show elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. Slight features of various phyla are described in chapter 4. 2.6 Viruses, viroids, prions and lichen. In the five kingdom classification of Whittaker, there is no mention of lichen and some acellular organisms like viruses, viroids, and prions. These are briefly introduced here. All of us who have suffered the ill effects of common cold or flu know what effects viruses can have on us, even if we do not associate it with our condition. Viruses did not find a place in classification since they are not considered true living. If we understand living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. Figure 2.6a TMV tobacco mosaic virus B bacteriophage are showing on diagram. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. Would you call viruses living or non-living? The name virus, that means we know more poison fluid, was given by as compared to new NCERT, it's given by M.W. Bejernik, not Pasteur, and not Dmitry Ivanovsky. Discovery was done by Dmitry Ivanovsky. Dmitry Ivanovsky in 1892 recognized certain microbes as casual organisms of the mosaic disease of tobacco. Figure 2.6a. These were found to be smaller than bacteria because they passed through bacteria-proof filter. M.W. Bejernik in 1898 demonstrated that the extracted of the infected plants of tobacco could cause infection in healthy plants and called the fluid as contagium vivum fluid, infectious living fluid. M.W. Stanley in 1935 showed that viruses could be crystallized or crystal consist largely of protein. They are inert outside their specific host cell. Viruses are obligate parasites. In addition to protein, viruses also contain genetic material that could be either RNA or DNA. No viruses contain both RNA and DNA. A virus is a nucleoprotein and the genetic material is infectious. In general, viruses that infect plants have single-stranded RNA, and viruses that infect animals have either single or double-stranded RNA or double-stranded DNA. Bacterial viruses or bacteriophages, viruses that infect the bacteria, are usually double-stranded DNA virus. Figure 2.6b, the protein coat called capsid made of small subunit called capsomer protect the nucleic acid. These capsomers are arranged in helical or polyhedral geometric forms. Viruses cause disease like mumps, smallpox, herpes, and influenza. AIDS in humans is also caused by a virus. In plants, the symptoms can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, dwarfing and stunted growth. Viroids. In 1971, Theo Diner discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be a RNA, free RNA. It lacked the protein coat that is found in viruses, hence the name viroid. The RNA of the viroid was of lower molecular weight. Prions in modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. The agent was similar in size to viruses. These agents were called prions. The most notable disease caused by prion are bovine spongiform encephalopathy (BSE), commonly called mad cow disease in cattle, and its analogous variant CR-Jacob disease (CJD) in humans. Lichens. Lichens are symbiotic association, that is mutually useful association between algae and fungi. The algal component is known as phycobiont and fungal component as mycobiont, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi and fungi provide shelter and absorb mineral nutrient and water from its partners. So close is their association, and if one saw a lichen in nature, one would never imagine that they had two different organisms within them. Lichens are very good pollution indicator. They do not grow in polluted areas. Summary. Biological classification of plants and animals was first proposed by Aristotle on the basis of simple morphological character. Linnaeus later classified all living organisms into two kingdoms, Plantae and Animalia. Whitaker proposed an elaborate five-kingdom classification, Monera, Protista, Fungi, Plantae and Animalia. The main criteria of the five-kingdom classification were cell structure, body organization, mode of nutrition and reproduction, and phylogenetic relationships. In the five kingdom classification, bacteria are included in kingdom Monera. Bacteria are cosmopolitan in distribution. These organisms show the most extensive metabolic diversity. Bacteria may be autotrophic or heterotrophic in their mode of nutrition. Kingdom Protista include all single-cell eukaryotes such as chrysophytes, dinoflagellates, euglenoid, slime mold, and protozoan. Protista have defined nucleus and other membrane-bound organelle. They reproduced both asexually and sexually. Member of kingdom fungi show a great diversity in structure and habitat. Most fungi are saprophytic in their mode of nutrition. They show asexual and sexual reproduction, Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes as the are the four classes under this kingdom. The Plantae includes all eukaryotic chlorophyll-containing organisms. Algae, bryophytes, pteridophytes, gymnosperm and angiosperm are included in this group. The life cycle of plant exhibit alternation of generation, gametophytic and saprophytic generation. They had the heterotrophic eukaryotic multicellular organism lacking a cell wall are included in the kingdom Animalia. The mode of nutrition of this organism is holozoic. They reproduce mostly by the sexual mode. Some acellular organisms like viruses, viroids, as well as the lichens are not included in the five kingdom system of classification. Thank you.